Good evening, children. Today we are going to discuss one of the important topic to know presence of starch in leaves. So before discussing of this topic, so subscribe the channel, share the channel, and like the channel. And biology classes are available in both the visible channel and also toppers class. So today our topic is what? To know presence of starch in leaves. It is one of the important topics in biology, state and NEET syllabus. So first, what are the required materials for this activity? A well-exposed potted plant, beaker, boiling tube, wire gauge. It is also called asbestos gauge, tripod stand, burner, methylated spirit, water, petri dish, iodine solution are the main required materials for this activity. So after collecting of all materials, then we'll go with the procedure. So what is the procedure of this activity? So first we need to take the leaf or fluffy leaf from well exposed plant to the sunlight. The plant which is well exposed to the sunlight, from that plant we need to pluck the leaf, fluffy leaf. And the leaf should be soft and thin. The leaf should be soft and thin. Pluck the leaf from well exposed potted plant to the sunlight, pluck a leaf, and it should be soft and thin, and put the leaf in a boiling water containing beaker. Is there any dust particles are there on the leaf? The dust particles will be removed by keeping up that leaf in boiling water. Then collect the leaf from boiling water containing beaker and put that leaf in a methylated spirit containing boiling tube. Next, place this methylated spirit containing boiling tube. Already we have inserted. Put that soft leaf in it in a methylated spirit. Then place this boiled tube in a water bath. So water bath. This is what water bath. The beaker containing water, and place the methylated spirit boiling tube. Already it is having leaf. Place that methylated spirit boiling tube in a water pot. So these are the required materials for how to arrange the apparatus while we are doing the experiment. We know that already we have discussed the required materials, and place that methylated spirit in a containing test boiled tube in a water pot. Okay, when we are applying heat to that, the water start boiling, and also leaf also inside the methylated spirit also start boiling, and the green color pigments, chlorophyll pigments will be removed. So for what purpose we are boiling the leaf in methylated spirit? To remove the chlorophyll pigments. So after 5 minutes, after boiling of that methylated spirit, so chlorophyll pigments will be removed from the plant, from the leaf, and collect the methylated spirit containing boiling tube from the water bath, and collect the leaf from methylated spirit by you with the help of forceps and wash with the water. Rinse with the water and place the leaf in a petri dish. Spread the boiling leaf. Spread it in a petri dish. Add 1 or 2 drops of iodine solution. Add 1 or 2 drops of iodine solution. After adding up iodine solution, the leaf color turns into blue-black color. Blue-black color. So after removing of boiling leaf from the methylated spirit containing boiling leaf, the color of the leaf is pale color. The pale color leaf should be rinsed with water and spread it in a petri dish. Add 1 or 2 drops of iodine solution. The leaf color turns into blue-black color, which indicate presence of starch in the leaves. Presence of starch in the leaves. So, observation. You know the aim of that activity to know presence of starch in the leaves. Required materials also we know. Procedure also we know. We have followed that. And now observation. So after boil the leaf, the color of the leaf turns into pale color, white color. Pale white color. After boiling the leaf in methylated spirit, color of the leaf turns into pale white color. Then next observation is what? After adding up iodine solution, the color of the leaf will be turns into blue-black color. Blue-black color. So these are the 2 points I have observed. We will also observe the same observation. So after boiling, the leaf color is very white color, and after adding of iodine solution, blue-black color. Okay, next conclusion. Conclusion or result. Result or conclusion. The development of blue-black color over the surface of the leaf, which indicate presence of starch in leaves. The development of blue-black color over the surface of the leaf, which indicate the presence of starch in leaves. It suggests that the leaves prepare their own food by the process of photosynthesis and then store the starch in leaves, within the leaves. So this is what to know the presence of starch in leaves. This is what is the aim of that? To know. How to know? To know about presence of starch in leaves. Presence of starch in leaves. Aim and required materials. Well exposed potted plant to the sunlight and beaker, water, and boiling tubes, methylated spirit, asbestos gas or white gas, tripod stand, burner, petri dish, and iodine solution dropper are the required materials for this activity. So once again we'll discuss the procedure of this activity. So first we need to take well exposed potted plant and pluck a leaf from well exposed potted plant, and it should be soft and thin. It should be soft and thick. Then boil the leaf. Keep that leaf or place that leaf in boiling water. Is there any dust particles on the leaf? The dust particles will be removed by placing of that leaf in boiling water. Then collect the leaf from the water, boiling water containing beaker, and place that leaf in a or insert in a methylated spirit containing boiling tube. For what we are keeping in methylated spirit? To remove a chlorophyll. Then glycolated spirit containing test tube. Already we have inserted leaf. The leaf should be placed in a water pot to remove a chlorophyll. After 5 minutes, so the water is start boiling the beaker, and also methylated spirit also start boiling. Now after 5 minutes, we need to collect the leaf from the methylated containing boiling tube and spread it in petri dish. Add 1 or 2 drops of iodine solution. Then the leaf color turns into blue-black color. When the leaf color is turns blue-black color, dark blue-black color, which indicate a presence of starch in leaves. So in the presence of sunlight, plants can prepare their own food and stored in a leaves to know the presence of starch in leaves. And what is the precaution while we are doing this activity? You know that boiled tube, methylated spirit containing boiling tube, it should not be directly place it or keep on the flame. It may catch fire. So that there are placing of that boiling test tube, methylated spirit containing boiling test tube, kept in a water bath. So otherwise it may catch the spirit, methylated spirit containing boiling tube, it may catch a fire. So this is the method of or procedure of presence of starch to know presence of starch in the leaves. So required material just we have discussed, and observation. After boiling, the color of the leaf will be paint white in color. After adding up iodine solution, the color of the leaves turns blue-black color. And conclusion or result is what? The development of blue-black color over the surface of the leaf, which indicate the presence of starch in the leaves. So this is our today topic, to know the presence of starch in leaves. So for the latest classes of biology, subscribe the channel, share the channel, and like the channel. Thank you. Thank you all.